Good afternoon. I'm Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled The Art and Science of Growing Animal Viruses in the Laboratory, Best Practices in Virus Culture, presented by Dr. Sujatha Rashid and Ms. Jessica Shiflett. Dr. Rashid is a virology scientist at ATCC. She received her PhD from the Christian Medical College in Vellore, India, where she also received training in medical and clinical virology. She has lead management oversight of the Virology Collection in Microbiology and Infectious Diseases Biological Resources Repository, MIDBR, also publicly known as BEI Resources, which is a government-funded biorepository managed by ATCC. Jessica Shiflett is a scientist and supervisor with 10 years of virology and assay development experience at ATCC. She earned her master's in microbiology at George Mason University, working with Dr. Wu. In the following webinar, they will discuss the basic principles for propagating animal viruses in the laboratory, with an emphasis on the art and science of virus culture that should be followed to help avoid contamination and cross-contamination. Further, they will provide an in-depth look at viral culturing practices in both cell culture and embryonated chicken eggs, using influenza virus as an example. If you have any questions for either speaker, please use the questions box available through the webinar program. Questions will be answered as time allows and will be archived with a recorded webinar presentation on the ATCC website at www.atcc.org.
ATCC was founded in 1925. We are a non-profit organization with a headquarters in Manassas, Virginia. We also have an R&D and a services center in Gaithersburg, Maryland. We are a world premier biological materials resource and standards development organization, and we hold about 5,000 cell lines, about 80,000 microbes in the collection, as well as several genomic and synthetic nucleic acids, and we also supply many media and reagents that support these reagents. ATCC collaborates with and supports the scientific community with industry, standard, and innovative biological solutions. We have a growing portfolio of products and services, and our sales and distribution are in 150 countries with 12 international distributors. Our team here is a group of talented individuals. We are about more than 450 employees, and over a third of them have advanced degrees. The outline of today's talk focuses on aseptic technique and best practices for avoiding contamination. We will go over culturing of viruses in tissue culture, followed by culturing of viruses in eggs. In tissue culture, we will touch upon choosing an appropriate host, cell line authentication, media and reagents used in virus culture, the importance of multiplicity of infection and defective interfering particles. We will also go over the steps followed in growing a virus in cells using the example of influenza virus. When we move on to culturing viruses in eggs, we will focus on what it takes to maintain the eggs, as well as a good source of the eggs that are used in virus culture. An example of how we grow influenza virus via the allantoic cavity inoculation will be explained, as well as hemagglutination assay. We'll finally end with viral preservation. Aseptic technique and avoiding contamination. Good aseptic technique is essential for avoiding contamination as well as cross-contamination. As not much emphasis is usually placed on teaching the art of cell culture, inadequate training has led to many failures. Common sources of contamination include poor laboratory technique, working with multiple cultures and cell lines concurrently, mislabeling of cultures, as well as using one reservoir of media for multiple cultures. The very first area that I want to explain on best practices is biosafety cabinets. First, we have to learn what is the biosafety cabinet we have in the lab and how it works to help maintain the primary containment. All biosafety cabinets must be certified when installed and whenever they are moved, and at least annually. Begin work by first checking the Magna-Haley gauge to ensure that it is working correctly, and keep a regular record of its function. Minimize the disruption of airflow by keeping only a small number of articles inside. This is because the Class 2 cabinets, which is commonly used, generally has requirements for protection of the product, the personnel, and the environment through the use of laminar airflow and HEPA filtration. So when you disturb the airflow, you will be compromising on containment. Most BSCs that we use in the lab have a front access opening that carefully maintain the inward flow. So when you move your arms in and out of the cabinet, do it slowly and perpendicular to the face opening to limit disruption of the air current. Place all the work materials inside before opening any container such as media or the virus culture flask, and limit the movement in and around the BSC to reduce airflow disturbances. Avoid blocking the airflow. By example, resting your arms on the front of the BSC will actually prevent the smooth flow of air in the grill that is immediately in front of you. Do not overreach for items and keep all of them within reach so you are not disrupting the airflow within the cabinet. Always perform the work about 4 inches back from the opening, and also use appropriate disinfectants to wipe all surfaces of the cabinet as well as any article that you take in and out of the cabinet before and after each use. For more details, the OSHA website has a fact sheet on laboratory biosafety cabinets which explains many of these in further detail. The next area where best practices should be followed is the media and reagents used. All reagents that you use for cell culture or virus culture, such as media, the PBS or trypsin, should be aliquoted and assign each aliquot to one cell line or one virus. This helps in preventing contamination as well as cross-contamination. Second, always use a new pipette each time going into a stock reagent. Do not reuse them because that's again another source of contamination. A common route of contamination of entry is by liquid bridge that forms when a droplet of culture media remains inside the culture vessel or its lid or on the neck of a culture medium bottle. So do not pour the reagents such as media, use pipettes. Do not move your arms, the hands or pipettes over open media containers, and keep these containers closed until they are in immediate use. For more on some of the practices that we follow when we are culturing cells as well as viruses in cell culture, first check the contamination of the seed virus that you're using prior to growing up a large stock. Contamination can come from, can be bacterial or fungal, so you can use a variety of media to see if something is growing in your seed stock before you put it out to cell culture. Preferably work only with one cell line or one virus at a time inside the biosafety cabinet, unless of course you're using an experiment that requires controls, such as a negative control or a cell control. When you're working inside the biosafety cabinet, work with non-infected material first, for example the cells, prior to any virus manipulation. And inside the biosafety cabinet, arrange items from clean to dirty in an orderly fashion, leaving a clear space to work in the middle. Keep uninfected cells and virus-inoculated cells in separate incubators, unless of course they belong to the same experiment. Always allow for clearance time between work to allow for air exchange. Generally speaking, we use about 15 minutes when we work with cell culture and about 30 minutes when we are working with a virus. So we are working with virus A, the work is done, give 30-minute clearance time before you work with virus B. Always remember to make sure your water baths and incubators are cleaned regularly, and have quarantine labs if it's possible in your facility, because when a material is known to be contaminated with bacteria or fungi, you would prefer to segregate and work with it in a different biosafety cabinet or a different room so that you're not contaminating other cultures that you may have going on in the lab. Personal protective equipment. The best practices is first you have to check what PPE requirements are required for the virus that you're working on. We also recommend changing gloves frequently while working with it inside the biosafety cabinet. When you're working, always wear a clean lab coat with long sleeves to prevent the introduction of contaminants from your arms or your clothing, and tuck the sleeves into the gloves as shown over here in this image. Do not spray disinfectant of the gloves, which we find is a common practice with people, because this actually will compromise the integrity of the glove. We find our best practice is to use disposable lab clothes when working with viruses. And when possible, because these are quite expensive, disposable sleeves is a cheaper option for people to use. Now moving on to the actual virus culturing. As you all know, viruses require living cells to support their replication. So oftentimes you have to use either a tissue culture system, or in certain cases an embryonated chicken egg or whole animal systems. With the utilization of cell culture technique, the use of laboratory animals has decreased significantly. And in comparison with eggs and animals, cell culture is more convenient and cost effective. Tissue culture systems generally comprise of cells that have originated from a multicellular organism, and they require special culture media that contain the essential nutrients and energy sources that are required for these cells to survive. It's a preferred type of growth system for many of the viruses. One point to remember about tissue culture or cell culture systems is that to ensure the viability, the genetic stability, as well as the phenotypic stability, cell lines need to be maintained in the exponential phase. So do not overgrow your cell culture in your flasks to the point that it is overgrown and crowded, that that cells will undergo a phenotypic change. Choosing a propagation host. Now, depending on whether you are using the cell line to isolate a virus from a clinical specimen, or whether you're growing a virus from a seed, you may have a few choices. From a clinical specimen, always pay attention to the host species and the permissibility of the host cells to that particular virus that you are expecting to isolate from the specimen. You have choices of using primary cells as well as cell lines, but remember that cell cultures vary greatly in their susceptibility to different viruses. So especially when you're trying to do isolation from a specimen having very small numbers of viral particles. Primary cells, while they have their advantages, they tend to be more permissive. They also have the disadvantage that many of these primary cells will carry adventitious agents. The most commonly used primary cell line in virus isolation is monkey kidney cells, and so there is the danger of any virus that is seen in the monkey that could also be present when you're trying to isolate the virus. For tissue culture adapted strains, ideally you want to keep the virus growing in the same host cell to minimize the selection of virus subpopulations. Oftentimes the genetic bottleneck, which is a selection event, will occur when a virus is passaged to a new host. If for your experiment needs there is a need to change to another host cell, ensure that the virus has adapted to it by checking for stable titers across several passages. Next, know about your host cell that you're using. First of all, are they authentic? Observe the morphology and viability of the cultures regularly and carefully. Macroscopic examination can give you some evidence of microbial contamination, but however, you need to be familiar with your cells. You want to use inverted microscope at low power, say 40x, and you want to check the medium for evidence of microbial contamination, as well as be familiar with how the cells look at different densities in the flask. Second, there is the issue of misidentified cell lines, which is nothing new to the scientific community. And there are two ways to detect this. One is the intraspecies identification by STR analysis. Now, ATCC has, as well as many other cell repositories, are beginning to use DNA polymorphisms in addition to other techniques such as enzyme polymorphism, HLA typing, and karyotyping to confirm the identity of their cell lines. One of the most reliable methods is the profiling of short tandem repeats, or STR, which is PCR amplification followed by capillary electrophoresis. STR profiles for all ATCC human cell lines are available on our website and the catalog descriptions. Interspecies identification is possible by an assay known as the cytochrome oxidase 1 or CO1 assay, which is used as a DNA barcode. The sequence of this gene is remarkably constant within a species but varies substantially from one species to another. It is also important to pay attention to the passage number of the host cell because the phenotype of highly passage cell line is quite different from their progenitor cells. Our routine practice at ATCC is to take a cell line from at the master seed bank and then grow it up to 10 passages when we use it for virus culture. Continuing on about authenticity of host cells, you also want to know if it is free of mycoplasma. And there are several methods to screen. Typically, cell lines are screened for mycoplasma by indirect methods such as using the Hoechst stain, which is fairly simple. The Hoechst stain usually uses a fluorochrome DNA stain that binds to the DNA of the mycoplasma, and the organisms are easily detected when examining using appropriate fluorescent microscope. We also have PCR-based detection as well as direct agar culture methods. The direct culture methods is quite long. It requires both agar and broth media to permit the isolation of cultivable strains, and they appear as characteristic colonies on agar medium. Finally, there is also I have to mention the influence of antibiotics and antifungal agents in media, which is a common practice in many labs. Our practice is to grow the cells in the absence of antibiotics because this will help us unmask the contaminants. Complete growth media. Complete growth media generally consists of a basal cell culture medium that is supplemented with other ingredients, ingredients such as serum, growth factors, there are trace elements and hormones. There are numerous formulations for the different cell types and the cell cultures have different requirements. The simple basic media, what we'd use for many of the cell lines and some of the more fastidious ones will require more complex as well as serum-free media. The type of medium used must be determined empirically for each media. While growing viruses, it's important to also know the difference between a growth media as well as maintenance media. Generally speaking, growth media has higher concentration of fetal bovine serum. Typically, it's about 10%. And this is what we would use when we are growing the cells prior to virus inoculation. The maintenance media, also more commonly we call it as the viral growth media, has a lower concentration of the serum, typically about 2%, and this is the media to be used once the virus is inoculated onto the cells. Here's an example of common media, Eagle's Minimal Essential Media, EMEM, and Delbeco's Modified Eagle's Medium, which is the DMEM. Be particularly cautious when you're working with a new cell line as media formulations vary among suppliers, even for media with similar or identical names. We suggest reading descriptions, formulations, and labels carefully to ensure that the appropriate medium is used or the cell line may be inadvertently adapted to the new medium. All ATCC cell lines come with information on their growth medium, and in most cases, the recommended medium and serum can also be purchased from the ATCC catalog. Also pay attention to whether the media contains HEPES buffer. Oftentimes, HEPES as well as other organic buffers can be used with many cell lines to effectively buffer the pH of the medium. In fact, some of these compounds that are used in the buffers can be toxic. We have seen that HEPES has been shown to greatly increase the sensitivity of media to phototoxic effects induced by exposure to fluorescent light. Antimicrobials. Now, routine use of antibiotics or antimycotics for cell culture is really not recommended unless they are specifically required. So when are they required? People have used it as a prophylactic to prevent contamination or as a cure once contamination is discovered. And other times, antibiotics are used for maintaining selective pressure on transfected cells. Antibiotics can mask the contamination of mycoplasma as well as resistant bacteria, and they can interfere with the metabolism of sensitive cells. Whereas antimycotics can be toxic to many cell lines and so should be avoided. We recommend that using the best practice of not using prophylactic antimicrobials for virus culture or even cell culture prior to virus inoculation. It is best practice, if you find that your cell line is contaminated, to discard and start over with new stocks, because particularly mycoplasma is very difficult to eliminate and you may actually lose your cell line. Fetal bovine serum. This is a rich source of growth factors and is appropriate for cell cultures and for growing viruses. The serum serves as a source of many important ingredients that are hard to replace, and primarily because of its rich content of growth factors and low gamma globulin content, FBS has been adopted as a standard supplement of cell culture media. There are different types of serum. Most commonly you use the filtered serum. These are used after filtering it through a 0.1 micron pore to remove adventitious agents. But you must sometimes, certain filters do not necessarily remove all of the mycoplasma or viruses. We also have gamma irradiated fetal bovine serum. This is the preferred serum to use, but it can be quite expensive. The irradiation inactivates viruses and other adventitious agents. Finally, you also have heat inactivated serum. ATCC does not routinely use heat inactivated serum unless specifically required for a particular cell line. Heat inactivation is generally detrimental to the growth of some cells because it reduces or destroys growth factors present in the serum. Originally, heat inactivation was performed to inactivate complement as well as to destroy mycoplasma contaminants. Removal of complement is usually quite unnecessary, but it can be important for certain assays and in cytotoxicity tests. Furthermore, there are lot-to-lot variations in fetal bovine serum. The exact composition of serum is unknown, and therefore it is a little difficult to get consistent results unless you are willing to use a particular lot for all your experiments to avoid the differences that may occur because of this variation from one lot of animal serum to another lot. Also, remember to check for endotoxin levels, especially if you're trying to isolate virus from clinical specimens, because that may actually deter from a good isolation. We also, in our experience, find that the country of origin is important, as there are regulatory implications depending from what country the serum originated from. And while all reputable suppliers test their products for infectious virus that may be present, there are instances when certain viruses are difficult to get rid of. As an example, there is foot and mouth disease. This is a USDA-regulated livestock pathogen. It's highly contagious. And so virus cultures that are grown in serum that originated from FMD-affected countries cannot be imported into the United States. The other agent to pay attention to is also the agent responsible for bovine spongiform encephalopathy, which is called the BSE. Unfortunately, there is no test for the presence of this agent, and so we recommend that you essentially obtain all your bovine products, including sera, from countries that are not affected by the BSE, so that this will help you for downstream use of the virus culture. Now, moving on to multiplicity of infection and its importance. Now, the multiplicity of infection, or MOI, it refers to the ratio of virus particles to infection targets, that is the host cells. Generally speaking, high MOI is used in certain experiments when they want to ensure that every cell in the culture is to be infected. And low MOI is used when multiple cycles of infection are required, as when you prepare a virus stock. And typically we use low MOI that range from 0.01 to 0.001, and this really depends on the virus that you are working on. Here's an example of how to calculate MOI. What you need is you need to know the virus titer as well as the number of cells that are in that flask that must be determined on the day of inoculation. Now, a question that comes about is multiplicity of infection more important or the virus concentration? And so for us, when we work at ATCC, when at the time of initiating an infection, to control for the variation of concentration with the volume of the inoculum used, we use small volumes during the adsorption step and always use the standard volume for a certain size of flask or dish that you would be working on. So the reason that we really pay attention to the MOI is because of defective interfering particles. These are virus-like byproducts. They're not infectious due to the presence of mutations in some of the essential viral genes. So they cannot replicate on their own. They are biologically active, and they are well known to have a huge impact on virus replication, the evolution, as well as pathogenesis. Even though they cannot replicate, when they co-infect cells with a viable intact virion, it can interfere with the growth of the virus. Obviously, the DI particles are more in RNA viruses because of the error-prone viral replication systems they have. They also interfere with having a good virus culture that will affect downstream processes. The best practice we have found is to use a lower MOI during cultivation of virus stocks. And by reducing the generation or amplification of these particles ensures that we have reproducible results as well as high yields and consistent product quality during virus production. Now we move on to initiating an infection in virus in tissue culture. So 1 or 2 days prior to virus inoculation, seed the host cells into at least 3 flasks. Typically speaking, if you're using one for virus growth, there should be another that controls for it, which is considered the mock infected. And then finally, a third flask is used for counting the cells on the day of infection. This is so that we can calculate what cells are there and do our MOI calculation. Avoid using cells that are seeded for more than 48 hours because you really want to use when cells are in their exponential phase. Allow the cells to reach the appropriate confluency for the particular type of virus you wish to grow, and this is usually should take place within 24 to 48 hours. And after you have calculated the MOI, you dilute the virus in a growth medium. So first, when you start with frozen viruses, you want to quickly thaw it in a 37-degree water bath and then dilute it in the viral growth medium. Rinse the cells prior to adding the virus inoculum, because sometimes for certain viruses particularly, the traces of FBS needs to be removed in order to have a good infection initiated. Some things to remember while harvesting a virus culture. First of all, the virus culture is incubated at the appropriate temperature and atmospheric conditions, and during the recommended incubation period, you have to observe the infected cells every day. You want to look for cytopathic effect as well as, if there's any possible contamination that can be observed macroscopically or even under the microscope. Determine the ideal harvest time, and for most lytic viruses, generally speaking, we harvest when the cytopathic effect has progressed to about roughly 80%. Do not let that CPE progress to a complete wipe out of the cells, because there will be a loss of titer. And during the harvest, depending on the virus, if it is a cell-associated virus, you can use freeze-thaw cycles to assist in releasing the virus, or some people have also used glass bead disruption of the monolayer. But however, do not use that for all viruses. It's particularly there are viruses that you would not recommend, for example, herpes viruses, because you would lose the titer.
As a working example of the guidelines Sujatha just covered, I'm going to talk through influenza growth in tissue culture. Both Vero and MDCK epithelial cells can be used to propagate influenza viruses in tissue culture. Despite being easier to work with, Vero cells do not support influenza A entry as well due to limited sialic acid, the receptor for influenza A. You can get better growth and titers of virus in MDCK cells, but these cells can be a bit more difficult to grow. Cells are split 1 to 2 days prior to infection using a seeding density that will allow the cells to reach the proper level of confluence for infection in that 1 to 2 day period. For influenza virus, a high confluence, about 95% of the flask surface area, should be attained. This high confluence is due to the virus growth media, which I will talk about on the next slide. When seeding flasks for infection, all the flasks are given the same density of cells. One flask is going to be used as the mock infection control, and that's used in contrast to the infected cells. One flask will be used for counting the total live cells immediately prior to infection in order to calculate the multiplicity of infection, or MOI. The total live cell number is multiplied by the MOI. An MOI of 0.01, for example, would mean 0.01 virus particles for each cell. The starting titer is a TCID 50 per mil, or the infectious dose per mil, is required to calculate this. Using an MOI at inoculation is important for generating infectious virus. If too much virus is added, defective particles or non-infectious virus are produced. Your mock control flask should be inoculated with the same volume of virus growth media as the inoculum volume added to the infection flasks. The reason that such a high cell confluence is required for influenza is because the virus growth media contains TPCK-treated trypsin, and this may cause some of the cells to dislodge from the monolayer. The virus growth media does not contain any FBS the way that we grow them here. It contains 1 to 2 micrograms of TPCK-treated trypsin, and trypsin is an endoprotease that cuts specifically on lysine and arginine residues. This cleaves the hemagglutinin of the influenza virus into 2 subunits to allow for virus attachment and entry into the cell. TPCK treatment inhibits protease activity of chymotrypsin, which is a common trypsin contaminant. Each lot of TPCK trypsin may have a different level of activity, and the final concentration of the trypsin is based on 1 to 2 micrograms of active units per mil. Influenza growth in tissue culture continued. Harvest the cells from the designated MOI flask and count live cells. Calculate the volume or dilution of virus inoculum required based on the TCID 50 titer. Thaw the virus stock in a 37-degree water bath until it is just melted, so that usually takes less than a minute. Prepare the virus dilution in virus growth media. Rinse the flasks designated for the mock infected control and then the flask set aside for infection with PBS. And then add the appropriate inoculum volume for your flask size, starting with your mock infection flask first, moving from clean to dirty, as Sujatha talked about. Rock the flask to evenly distribute the inoculum, and then incubate for the virus absorption time. For influenza virus, 33 degrees is typical for influenza B, and 35 degrees is typical for influenza A in 5% carbon dioxide atmosphere. Absorption is typically 45 minutes to an hour. After the absorption period, feed the flask with virus growth media, and that will depend on the size flask you're using, and incubate for the recommended time. Most influenza viruses grow within 2 to 3 days. Monitoring the infection and the mock infected control daily is important in order to recognize early cytopathic effect. Some of the cytopathic effects, or CPE, are very subtle, so it's really important to compare it to your mock or negative control. Harvest when CPE is evident in approximately 80% of the flask. Here is shown a picture of CPE caused by influenza A in MDCK cells. It causes refractile rounding, where cells appear shiny and detach from the monolayer. If you look at the negative or mock infected flask, refractile rounding can also be seen as cells grow, especially when they're over confluent. So as infections go on, you may see more of this in your mock infected control. That's why it's so important to recognize CPE in comparison to your mock infected flask. In cases where you have a virus that does not cause CPE, a fluorescent antibody can be used to detect the virus. In the case of influenza, you can also do an HA assay, which I will talk about a little bit later. For a fluorescent assay, you use a pipette and scrape a small area of your flask and transfer infected cells to a slide. If the antibody assay does not show a good proportion of infected cells, you continue the virus incubation, and then you would retest until you got a good proportion of cells infected. All right, virus can also be grown in eggs. They're convenient and an expensive host for many viral strains. High titers can be reached. There are several membranes and cavities within the egg that can support viral growth. The inoculation site depends on the growth requirements of the specific virus. Virus growth can cause embryo death, the embryo cell damage, lesions on the egg membrane, or agglutination of red blood cells. Here are some examples of other viruses that can be grown in chicken eggs. Herpes simplex virus and vaccinia virus can also be grown in eggs, and different parts of the egg can be used for propagating virus. Influenza A's and B's are most commonly grown in allantoic fluid. Some important things to consider when using eggs are chicken eggs should be obtained from flocks that are confirmed to be free of specific disease pathogens. Upon arrival, eggs should be incubated at 37 degrees in an atmosphere of 40 to 70% humidity to ensure proper development of the air sac prior to virus inoculation. Eggs should be candled a couple of hours after arrival to ensure that embryos are viable and properly developed. For candling the eggs, you hold the blunt end of the egg against a light source and use a pencil to outline the air sac, which appears transparent. Use a pencil to also mark the embryo eye spot, which appears as a dark spot. Sometimes you have to slowly turn the egg to get the eye spot to show up. The air sac is indicated for a guide for infecting allantoic fluid with influenza virus. Influenza A and B viruses should be inoculated into 9 to 11-day-old eggs. Here's an example of candling eggs. On the far left, you have an 11-day-old fertilized egg. You can see that air sac at the top of the egg that appears transparent. The blood vessel network is healthy and intact, and you can also see embryo movement in response to the light from candling. The embryo is tightly surrounded by the amniotic cavity, and that's attached to a yolk sac. The allantoic cavity is usually situated above or to the side of the embryo and is separated from the air sac by the allantoic membrane. In the middle is a non-viable embryo. You can see some collapsed blood vessels above the yolk sac, and there's no embryo movement. On the far right, there's an unfertilized egg. There's no blood vessel network visible. There's a large yolk sac, and there's no embryo. Here's another photograph of a non-viable embryo. Notice the collapsed blood vessels circled in the middle. All right, all work with virus should be done inside a Class 2 biosafety cabinet wearing the proper PPE, for example, gloves, a lab coat with long sleeves like Sujatha mentioned, safety glasses. So preparing the influenza virus, you're quickly thawing that in your 37-degree water bath and diluting it in PBS to approximately 1,000 CEID 50 per egg. That's chicken egg infectious dose. You wipe the eggs with an ethanol-soaked sterile cotton. Do not spray the eggs with alcohol, as that can change the permeability of the eggs and actually allow for more infection of bacterial contaminants. You pierce the hole on the opposite side of the marked embryo eye spot. So inside the line that you drew for your air sac of the chicken egg, on the opposite side of the X where marking the embryo spot, using a 18-gauge needle, you poke a hole in the eggshell. Make sure you're not holding the egg when you do this. Keep it in the tray so that you don't inadvertently stab yourself with the needle. And then after you've poked holes in all of the eggs, you get a 22-gauge needle attached to a syringe with your inoculum. First, you're going to inoculate your negative control eggs with just plain PBS, and then you will move to inoculating your eggs set aside for virus growth. Using the 22-gauge needle attached to a syringe, you should have your needle straight up and down, and you will enter the needle approximately halfway of the needle's length into the egg in order to reach the allantoic cavity. You may feel slight resistance or piercing of the allantoic membrane, and you dispense 0.2 milliliters of virus per egg, which you've calculated to be 1,000 CE-50 per egg. Needles are a sharps hazard and should be rinsed with disinfectant such as 10% bleach and disposed in a biohazard sharps container. Do not recap needles. After you've inoculated all of your eggs, seal the hole with a drop of nail polish. Nail polish is used rather than wax or glue because it contains a little bit of acetone, which helps prevent bacterial growth. Once again, you're going to rinse the top of each egg with an ethanol-soaked cotton swab. And then when the nail polish is dry, you move your eggs into the incubator and incubate under conditions appropriate for viral replication. Candle the eggs 18 to 24 hours after inoculation. Discard any eggs that are non-viable due to needle stick injury. At the end of the incubation period, refrigerate eggs for at least 2 hours. This will help constrict blood vessels and prevent blood from contaminating your virus and your allantoic fluid when you harvest. Using the Biosafety cabinet, wipe the eggs with ethanol-soaked cotton again. Open the egg by gently tapping on the shell just above the air sac until the shell breaks. Use sterile scissors or forceps to cut away the shell around the air sac. Keep your scissors cutting inside the line that you drew so that you don't accidentally cut into your yolk sac or your allantoic fluid. Using a sterile pipette, poke through the allantoic membrane into the allantoic cavity. It actually works out best if you just do one motion. If you bring the pipette in and out many times, it will cause contamination, and it will also cause that allantoic membrane to clog your pipette. So if you can get it the first try, great. If not, get a new pipette because the one you tried the first time will probably be clogged. Slightly tilt the egg to allow the allantoic fluid to pool on the side of the egg, and then you aspirate your virus in the allantoic fluid with your serological pipette. As you're pulling up the allantoic fluid, check for obvious signs of bacterial or fungal contamination. It should be clear allantoic fluid. If there's egg yolk or blood, it can cause false negative results by your hemagglutination assay. But if you have blatant bacterial contamination, the allantoic fluid will appear cloudy or sometimes a greenish yellowish color. If you notice that in an egg, harvest that fluid separately so that it does not contaminate your pooled virus. Approximately 5 to 10 milliliters can be harvested from each egg when you're planning on how much virus you're trying to grow. If you do get yolk or blood contaminating your virus, spin clarify your virus prep prior to your HA test to avoid false negatives. After virus is harvested from eggs, it may be pooled, again leaving out anything you suspect contamination. Eggs are treated as biohazard sharps. After harvest, eggs should be put in double-bagged biohazard waste inside a metal pan. It is also a good idea to keep these harvested eggs inside a refrigerator until they can be autoclaved. The hemagglutination assay is to test for the presence of virus. In a 96-well U or V bottom plate, add 100 microliters of uninfected allantoic fluid that's harvested from your negative control eggs, or your virus, or a positive control, which can be any virus that's previously shown positive HA results. Add 100 microliters of 0.5% chicken red blood cell solution to each well, and allow the plate to sit at room temperature for 30 minutes. After 30 minutes, a positive HA result forms a lattice at the bottom of the well. Red blood cells attach to virus particles, preventing the red blood cells from accumulating at the bottom of the tube. Red blood cells stack up in a slightly overlapping arrangement, making the bottom of the tube look fuzzy, and that is the lattice, which we refer to. A negative result is seen as a round button of red blood cells sinking to the bottom of the tube. To determine the titer, titer is expressed as a reciprocal of the highest dilution of virus that causes a lattice formation with RBCs, or red blood cells. The titer for eggs is expressed as egg infectious dose, and the method of Reed and Munch is used for calculating an endpoint titer.
To conclude, we will speak a little bit on the viral preservation methods. There is really no single procedure that's optimal for preserving all animal viruses. It is important to remember that for cell-associated viruses, slow freezing is recommended to achieve a cooling rate of -1 degree C per minute. People have used freezing media, which you can use, is essentially media containing cryoprotectants such as glycerol and DMSO. And for the viability of viruses that are not cell-associated, it is best maintained by rapid freezing, in which case viruses are quickly frozen in a dry slurry or liquid nitrogen vapor. For storage, we recommend you can store either in liquid nitrogen vapor or within a mechanical freezer, which is maintained at -80 degrees Celsius. And for very long-term storage, ultra-low temperatures, that is below -120 C, are required. There are two materials to choose from for cryopreservation vials. Either you can use glass or plastic. Glass vials are more difficult to work with. They need to be sterilized before use, and they generally do not come with labels such as information that is imprinted onto the side of a glass. They need to be sealed with a hot flame, and they can be quite difficult to open. More oftentimes, it is the cryo vials that are used, and that is what we would recommend. So to summarize, we have learned that virus culture is quite complex because of how cell cultures have to be maintained, and also the nature of biomaterials that are used in maintaining these cell cultures and virus cultures complicates the process of growing good viruses. To summarize, we have seen that it is good laboratory practices and aseptic technique which are essential for properly growing and maintaining virus stocks. When propagating the viruses in tissue culture or cell culture, choose the ideal propagation host. Ensure the host cell is authentic. Use the correct MOI and media that are required for virus cultivation. When you're propagating viruses in embryonated chicken eggs, use SPF eggs, that is specific pathogen-free eggs, and ensure that each egg contains a viable embryo and is healthy prior to inoculating with virus. There is really no single procedure that is optimized for virus preservation, and you have to determine this depending on the virus you have, the virus that you're growing. So what we would recommend is to control the steps and processes where you can by using the best practices that we have mentioned in this webinar.
Thank you, Dr. Rashid and Ms. Shiflett, for that excellent presentation. In just a few moments, we will begin our question and answer session. Please use the question box available through the webinar program to submit questions. And this session will be archived along with a recorded webinar presentation on the ATCC website at www.atcc.org. So for our first question, how can you differentiate between CPE and cytotoxicity?
This is certainly difficult for those who are new to virus culture. But remember that viral-induced cytopathic effects in host cells are caused by lytic virions. So you can always check for the early signs of cytopathic effect, which generally begins in small foci in the monolayer before they spread across. In contrast, cytotoxicity tends to appear throughout the monolayer. So when you use suitable controls such as mock-infected controls that has been treated in the similar manner as the virus-infected flasks, you will be able to see the differences. Cytotoxicity can generally be eliminated or reduced by dilution or filtration prior to the actual inoculation of the virus cultures as well.
Our next question is, how do you pick a host cell line for something you've never grown before, such as newly emerged virus in a clinical specimen from a suspected case?
I would suggest doing a literature search to see how members of the virus family were propagated to kind of narrow down the appropriate cell lines. Also, you need to be familiar with the kind of disease and what is the specimen that is coming from this particular suspected case, and suggest that you inoculate 2 to 4 different cell lines so that you can get a good chance of isolation from the virus at least growing in any one of these.
Our next question is, where can I send my cell lines to be checked for cell line authentication?
ATCC offers 2 services for cell line authentication. One is the STR profiling, as I mentioned earlier. This helps to detect misidentified, cross-contaminated, or even genetically drifted cells, which tends to invalidate many research results. And the other service we offer is the mycoplasma testing, which we do by two methods, the stain as well as the direct culture method.
Our next question is, how do I check for mycoplasma contamination of cultures in my lab?
There are several PCR-based detection assays that are commercially available, and you can perform the test yourself. ATCC also offers the universal mycoplasma detection kit, which you can purchase via our website.
Our next question is, how do I get rid of mycoplasma contaminants in my virus cultures?
There are many reagents that are available. Generally, they're a combination of antibiotics, or sometimes there are certain chemicals that can affect the mycoplasma. Just remember to treat your virus culture for 2 to 3 passages with whatever agent you're using, and then passage another 3 times in the absence of the agent to ensure that the mycoplasma is truly eliminated. You can also treat with ether, which removes mycoplasma, but this can be only done for ether-resistant strains. And finally, if you have to clean a virus culture that is a virus which is a positive-stranded RNA virus, you can extract the viral RNA from the culture. The extraction process, the lysis buffer during the extraction process will get rid of the mycoplasma, and then you can transfect that viral RNA into the appropriate cell line. This method generally helps to avoid passaging of the virus. However, we can only do it for positive-stranded RNA viruses that are, as an example, flaviviruses or alpha viruses.
So we'll take one more question, and that is, how do you get rid of DI particles in the virus seed?
So the DI RNAs generally retain replication and packaging signals, and they tend to be synthesized preferentially over infectious genomes. And so one way of trying to get rid of them is very simple. It's based on limiting dilution. So essentially you can dilute out the DI particles. This is a little time consuming, and you will have to spend a few passages to bring back the virus titer. But when you do that, you will eliminate the DI particles, and you will be left with good virions, and that will end up being a good seed material for future work.
So at this time, we'll conclude our question and answer session. I would like to thank the speakers for their excellent presentation, and thank you everyone for attending the webinar. Please join us for more webinars in the ATCC Excellence in Research webinar series. You can register for our upcoming webinars at www.atcc.org/webinars. And for more information about viral propagation, please download a free copy of our ATCC Virology Guide at www.atcc.org/guides. Thank you again, and have a great day.